Imagine stepping out of your front door right now. The moment you cross the threshold, you are instantly crushed by the equivalent weight of 50 fully loaded jumbo jets pressing down on every single square inch of your body.
Which is just, an unfathomable amount of physical pressure to even conceptualize.
Right. And simultaneously, you are plunged into absolute pitch black darkness. A void so complete that your eyes actually strain just trying to comprehend the total absence of light.
And we can't forget the temperature, which plummets instantly to just a microscopic fraction of a degree above freezing.
Exactly. You are now standing in the largest, most pervasive habitat on planet Earth. Welcome to Audio Notebook. I'm so glad you're here with us today because our mission is to explore the roughly 70% of Earth that remains our planet's greatest and, honestly, most terrifying mystery: the global ocean.
When you frame it with those physical constraints, the cold, the pressure, the dark, it really reorients how we think about our own planet. We casually call it Earth, primarily because dirt and rock are what we physically walk on every day.
Yeah, we have a very terrestrial bias.
We really do. But objectively, if you observe our world from space, we live on planet Ocean. If we calculate the actual volume of space where life can actively exist, that habitable biosphere, the ocean constitutes more than 90% of it.
More than 90%. That is just staggering.
It is, and yet, despite being the defining feature of our world, it is by far the least understood, least explored major habitat in existence. We are essentially strangers in our own home.
I really want to unpack this right from the jump because the sheer scale of what we don't know is so difficult to wrap my head around. You always hear that popular cliche, right? We know more about the surface of the moon than the bottom of the ocean.
Right, people say that all the time.
But when you just laid out those physical realities of the marine environment, it suddenly makes perfect sense.
Yeah.
On the moon, there is no atmosphere, so you just have a vacuum. In the ocean, every 10 meters you descend, the hydrostatic pressure increases by 1 full atmosphere.
It's an incredibly aggressive environment.
It is. You have that crushing pressure, the near freezing cold, the absolute darkness once you drop below the sunlit zone, and this highly corrosive saline fluid surrounding everything. It is an environment that actively, almost violently, fights human observation.
That physical barrier is really the crux of the entire problem. Historically, it has rendered the deep ocean completely inaccessible to all but the most heavily engineered, extraordinarily expensive human endeavors.
Which means most scientists just couldn't get down there.
Exactly. Because of this extreme inaccessibility, the scale of human ignorance regarding marine biodiversity is profound. The current scientific consensus suggests a pretty staggering reality, which is that fewer than 10% of the species inhabiting the ocean have been formally documented.
Less than 10%. That is wild.
And the urgency here cannot be overstated. Right now, as we speak, there is a literal race against time happening beneath the waves.
A race between discovery and destruction, basically.
Exactly that. On one side of the ledger, you have groundbreaking artificial intelligence and cyber taxonomy racing to discover and categorize this invisible world. And on the opposing side, you have global industrial mining conglomerates actively preparing to strip mine the abyssal plains.
So we are essentially trying to inventory a massive warehouse in the pitch dark, while the bulldozers are literally warming up in the parking lot.
That is, unfortunately, a very accurate way to put it.
To truly understand the mysteries of the deep, I feel like we first need to establish exactly how much we don't know, which I realize as I say it out loud sounds like a complete paradox. Like, how do scientists count things they haven't actually found yet?
It is a massive macroecological debate, and the methodology behind it is actually fascinating. So a landmark study was published in 2011 by a researcher named Mora and a team of colleagues. They set out to calculate the total number of species on Earth.
But without having the actual specimens in front of them.
Right. To do this without the physical animals, they didn't just throw out a random guess. They utilized something called asymptotic patterns. Essentially, they analyzed the predictable mathematical scaling patterns of higher taxonomic classifications.
Okay, let me pause you there for a second because asymptotic patterns is a pretty heavy phrase. If I remember my high school math correctly, an asymptote is a line on a graph that a curve gets closer and closer to, but it never actually touches. How does that apply to counting crabs and fish?
The best way to visualize it is to imagine the tree of life. The massive, thick branches at the very bottom are the phyla, so broad categories like arthropods or mollusks.
Okay, I'm picturing it.
As you move up the tree, the branches split into smaller and smaller twigs. Class, order, family, genus, and finally, the individual leaves at the very top represent the specific individual species.
Got it. The leaves of the species.
Historically, scientists have been very good at identifying those big branches. The discovery rate for new phyla or classes leveled off decades ago. The curve flattened out. It reached its asymptote.
Oh, I see.
So, by tracking the mathematical relationship between the known big branches and the known smaller branches, Moore's team could extrapolate those mathematical curves all the way down to the species level.
So if you know exactly how many thick branches there are, and you understand the specific mathematical ratio of how those branches split, you can mathematically predict how many leaves must exist at the very canopy of the tree.
Precisely. Even if that canopy is hidden in the clouds and you can't physically see the leaves.
That is a brilliant way to model it. So what did the math tell them?
Using those mathematical projections, the 2011 study predicted that there are approximately 8.7 million total eukaryotic species globally, meaning organisms with complex cells, so excluding bacteria.
Okay, 8.7 million total. How many in the ocean?
Of those, their models indicated that roughly 2.2 million inhabit the marine environment. But the most alarming part of the paper was the implication of that number. Based on the catalog of species we actually had on file at the time, a 2.2 million total meant that an astonishing 91% of marine species remain completely undiscovered.
91%. I mean, that means for every single fish, crab, sponge, or jellyfish we have documented, there are 9 more just swimming or crawling around down there, completely off the books.
Which is a daunting prospect for any biologist.
But, knowing how the scientific method works, I have to imagine a claim that massive didn't go unchallenged. Science rarely just settles on the very first model.
No, absolutely not. The pushback from the taxonomic community was swift and significant. The very next year, in 2012, an exhaustive synthesis was published by a researcher named Appletons and his team.
What was their approach?
They compiled the first truly comprehensive register of the world's known marine life, called the World Register of Marine Species, or WORMS for short.
Good acronym.
Very fitting. They established a firm baseline counting about 226,000 described eukaryotic marine species, but then they introduced several critical variables that completely tempered that massive 2.2 million estimate from the previous year.
And this is where the story gets highly relatable for anyone who is disorganized, because a huge part of the problem with our historical data was just really sloppy record keeping over the last couple of centuries, right?
The history of taxonomy is incredibly messy, yes. The Appletons researchers had to account for a massive accumulation of synonymy in biological nomenclature. To put a hard number on it, they identified approximately 170,000 synonymous names cluttering the historical scientific record.
I genuinely love this detail. It's exactly like looking at your smartphone and realizing you have 10 different contacts for the exact same person.
Oh, perfect analogy.
You might have, John Work, John Jim, John Smith, and John from the party. They're all the exact same guy. You just met him in different contexts and gave him different labels.
Except in marine biology, it's a 19th century French scientist finding a crab and naming it, and then 20 years later, a British scientist pulls up the exact same crab.
And maybe thinks it looks slightly redder because of the lighting on his ship, so he gives it a brand new, formal Latin name.
Exactly. The scientific literature was completely riddled with that scenario. Hundreds of thousands of supposedly new species were simply misidentified duplicates of existing ones. So the 2012 team aggressively subtracted those duplicates from the equation.
Okay, so that shrinks the number down. What else did they adjust?
They incorporated another fascinating, often overlooked variable, the immense backlog of physical collections. Based on surveys of global institutions, they estimated that between 58,000 and 72,000 marine species have actually already been collected.
Wait, we have them.
They physically exist in human possession, but they are sitting in glass jars of formaldehyde in the dusty back rooms of museums and university archives around the world, just waiting for an expert to formally examine and describe them.
That is wild to picture. Tens of thousands of unknown alien creatures are just gathering dust in a basement somewhere. So they've technically been caught by a net, but they aren't official to science yet.
They are trapped in a bureaucratic limbo, essentially. Finally, the researchers adjusted their models to account for cryptic species.
What makes a species cryptic?
It introduces a layer of genetic complexity. Cryptic species are organisms that are morphologically identical, meaning they look exactly the same to the naked eye and sometimes even under a powerful microscope, but they are genetically distinct.
Oh, so you can only tell them apart by looking at their DNA.
Exactly. They can only be differentiated through advanced molecular DNA sequencing. When the Appleton's team ran all these adjustments, subtracting the duplicates, adding the museum backlog, and calculating the genetic cryptic variations, their 2012 study projected a heavily revised global total.
And what was the new number?
They estimated between 0.7 and 1.0 million marine species total.
Okay, so the great taxonomic debate essentially boils down to this.
Yeah.
Is the planetary inventory roughly 1 million, or is it way over 2.2 million?
That is the dividing line in the literature.
But honestly, when you take a step back, what does this actually mean for us? Because regardless of which camp of scientists is mathematically right, the fundamental conclusion is identical.
Which is?
Between 1/3 and 2/3 of all complex marine life on Earth remains entirely unknown to science.
Yes. The empirical field data backing up that massive gap is incredibly consistent. If you analyze recent deep ocean and coastal surveys globally, the average find rate is remarkable.
How remarkable are we talking?
Approximately 37% of the species encountered during these expeditions are completely novel. You drop an autonomous camera or take a sample, and more than a third of what you pull up has never been seen or documented by human eyes in the history of our species.
Just try to map that onto our everyday experience. Imagine walking into a local forest or a park, and a full third of the birds landing in the trees, or a third of the insects crawling on the ground, are creatures entirely unknown to science. We wouldn't accept that level of ignorance in our own backyard.
We absolutely wouldn't.
So why is it taking so long? Why can't we just, hire more biologists and speed up the cataloging process?
The bottleneck you are describing is known in the field as the taxonomic impediment. The core issue is that the traditional workflow for biological taxonomy still heavily relies on methodologies established in the 18th century by Carl Linnaeus.
Still using 18th century methods for a 21st century problem.
Right. To officially name a new species the old-fashioned way, you first have to physically capture a pristine, intact specimen. Then, a highly specialized expert must conduct a meticulous physical morphological examination.
Like counting scales and measuring fins.
Exactly. Measuring every single spine, scale, and fin ray. Then they have to write a detailed manuscript, submit it to an academic journal, navigate the peer review process, and finally get it published.
That sounds exhausting. How long does that whole pipeline take?
When you average out this entire bureaucratic and scientific pipeline, it currently takes 13.5 years from the moment a marine specimen is collected in the ocean to the day it is formally granted a name.
13 and a half years. That is agonizingly slow, especially when you consider that the ocean is warming, it's acidifying, and the chemistry of the water is fundamentally shifting.
It's shifting much faster than we can document it.
Exactly. We are effectively trying to document a burning library by meticulously copying the text of each book by hand.
The burning library is an incredibly apt way to frame the tension of everything we are looking at today. Because to truly comprehend the magnitude of what we risk losing, we have to transition our perspective a bit. We have to move from the abstract, mathematical numbers of what we don't know into the physical reality of the ocean's interior. We need to examine a massive, tangible biological phenomenon that, for a significant portion of modern history, humanity completely misunderstood.
Oh, this is one of my favorite pieces of scientific history. We are talking about one of the greatest technological mix-ups ever recorded, and it's a story that actually starts with the military.
It does. It's a fascinating pivot.
Let's set the scene for you. It's the summer of 1942. World War II is raging across the globe. Off the coast of San Diego, California, there's a naval research ship called the USS Jasper. On board is a specialized team of acoustic physicists, led by a scientist named Carl F. Ehring.
And to be clear, their mission has absolutely nothing to do with marine biology.
Nothing at all. They're out there testing this brand new, highly classified cutting-edge technology called long-range active sonar. Their singular goal is to detect the presence of Japanese submarines and German U-boats lurking in the Pacific.
The mechanics of active sonar involve emitting powerful acoustic pulses pings of sound down into the water column. Then, the operators precisely measure the time it takes for the echo to bounce off a dense object and return to the ship.
They were listening for the hard, unmistakable acoustic return of a steel submarine hull.
Right. They send the ping down, waiting for an enemy sub, but instead they hit a wall. About 300 yards below the surface, their instruments registered this massive, unyielding, dense anomaly.
It was scattering their acoustic signals so thoroughly and so consistently that it appeared on their sonar screens as the actual bottom of the ocean. They thought they had found a shallow, false ocean floor just floating in the middle of the Pacific.
If you consider the context of naval warfare, having a phantom ocean floor suddenly appear on your instruments is a tactical nightmare. It completely blinds your ability to detect anything below it.
But what made this anomaly truly baffling to the physicists wasn't merely its existence, it was its behavior.
Right, because a mountain or a seabed doesn't move.
Exactly. But every single evening, as the sun began to set and twilight descended over the Pacific, the sonar operators watched in disbelief as this phantom seabed rose hundreds of meters toward the surface, creeping closer and closer to the hull of their ship.
And then, like clockwork, every morning as the sun came up, the phantom seabed would sink back down into the black abyss.
They officially named it the ERC layer after the three scientists who documented it, Ehring, Rate, and Christensen, but they had absolutely no idea what they were looking at.
The phenomenon remained a classified military mystery until 1945. That's when a biological oceanographer named Martin W. Johnson, who was collaborating with the University of California Division of War Research, proposed a radical, almost unbelievable hypothesis.
What do you think it was?
He suggested that this impenetrable acoustic layer wasn't a physical temperature gradient or a saline density change or some bizarre technological glitch. He hypothesized that the false ocean floor was entirely alive.
A living, breathing ocean floor.
Yes. To prove it, Johnson conducted a series of overnight excursions off Point Loma, California. He didn't use sonar. He used depth-stratified net hulls. He dropped physical nets precisely into the depth zones where the acoustic anomaly was registering, tracking it as it moved up at night and down in the morning.
And what did he pull up?
His samples provided the undeniable proof. This false bottom was actually composed of billions upon billions of individual marine animals undertaking a highly synchronized daily vertical migration.
Today, marine ecologists refer to this phenomenon as the deep scattering layer, or the DSL.
And that daily commute they do is called DDM diel vertical migration.
The environment they inhabit during the day is the mesopelagic zone, famously known as the twilight zone. This zone stretches from about 200 meters down to 1,000 meters. If you were floating in it, you'd experience rapidly diminishing, faint blue light, intense pressure, and dropping temperatures.
It's a harsh transition zone.
So I have to know, who is actually participating in this massive migration? If you were to dive into the deep scattering layer right now, what kind of creatures are brushing past your face?
You would be surrounded by a highly adapted, incredibly diverse community of micronectin and zooplankton. The most prominent vertebrate residents are mitophids, which are commonly known as lanternfishes. You also have bristlemouths.
What exactly does a bristlemouth look like?
They are small, slender fish, typically only a few inches long, with jaws full of needle-like teeth, hence the name. But what's astonishing about bristlemouths is their sheer population density. They are widely considered by biologists to be the single most abundant vertebrate genus on the entire planet.
More than any bird, rat, or human.
Far more. Alongside the fish, you have vast numbers of cephalopods, including various agile squid species, and then you have a massive, often underappreciated component of gelatinous life.
Like jellyfish.
Yes, and pelagic tunicates like pyrosomes, which look like glowing, hollow tubes and salps, accompanied by massive swarms of crustaceans like krill and microscopic copepods.
Because they live their entire lives in this dim twilight zone, they have evolved some of the wildest adaptations in nature. Since there's barely any sunlight, they possess highly sensitive visual systems. Some of these creatures have eyes so advanced they are capable of detecting single, solitary photons of downwelling sunlight.
Their vision is pushed to the absolute physical limits of physics.
And then there is the bioluminescence. They literally glow in the dark. They use it for intraspecies communication, to attract mates, and brilliantly, they use it for something called counter-illumination camouflage.
That is a phenomenal adaptation.
It really is. They have light-producing organs on their bellies that precisely match the faint blue glow of the surface water. So if a predator is swimming below them and looks up, the twilight zone fish completely blends in with the light from above. It is biological cloaking technology.
It is a masterpiece of evolutionary engineering. But here is the critical historical turning point regarding the DSL. For decades after its discovery, accurately measuring the sheer biomass, the total physical weight of all these animals combined, was one of the greatest, most frustrating challenges in marine ecology.
Why was it so hard to weigh them?
Because our traditional methodologies were fundamentally flawed. We relied almost entirely on dragging large nets through the water column.
And using nets to catch the twilight zone is like trying to catch a flock of birds by dragging a chain link fence behind a tractor.
That is a very visceral but accurate image.
Well, first off, a research ship dragging a massive midwater trawl generates a tremendous amount of disturbance. You have the visual cue of the net, the loud acoustic noise of the cables and winches, and a massive pressure wave pushing through the water ahead of the net itself.
All the fast-swimming animals in the layer, the squids, the agile lanternfish, they feel that pressure wave coming and simply dart out of the way.
That is the avoidance bias.
Exactly. Then you have the destruction bias. The fragile, gelatinous organisms, the pyrosomes, the salps, the jellyfish, which we now know make up a massive integral portion of the layer's biomass, cannot swim away.
They just get caught in the mesh.
They are swept into the net, yes, but because they are so delicate, the intense hydrodynamic pressure of the trawl being dragged for hours literally pulverizes them. They are turned into unrecognizable organic mush and washed right out the back of the net, completely lost to the scientific record.
So the fast ones swim away and the squishy ones get obliterated. Because of these twin failures of nets, early ecological estimates were incredibly dangerously low. For a long time, the accepted scientific consensus suggested that the global mesopelagic fish biomass was approximately 1 billion metric tons, or 1 gigaton.
Which is still a huge number, but...
Right, I mean, 1 billion metric tons still sounds like a staggering amount of life until you realize just how wrong the nets were. To fix the error, scientists had to undergo an acoustic revolution. Instead of relying on physical nets, they went back to where the story started: sound, but with vastly superior technology than the WWII sonar.
Precisely. The advent of broadband acoustic Doppler current profilers, or ADCPs, and sophisticated multi-frequency echo sounders completely revolutionized our understanding of the midwater ecosystem.
How is it different from the WWII pings?
Instead of a single blunt ping of sound, modern oceanographers utilize a spectrum of distinct acoustic frequencies. By analyzing how different frequencies bounce back, they can essentially categorize the major biological constituents of the DSL based entirely on the physical composition of the animals' bodies.
This is where the physics absolutely fascinates me. I'm trying to visualize this. If I yell into a canyon, I just hear an exact echo of my own voice. It doesn't tell me if the canyon wall is made of granite or sandstone.
Mm-hmm.
How does a scientist shoot a sound wave into the dark ocean and know, that's a swarm of lanternfish, and right below it is a swarm of krill?
It fundamentally comes down to the physics of acoustic resonance and the density of the target. Think of different frequencies of sound like different tools. Lower frequencies, such as 18 kilohertz and 38 kilohertz, have longer wavelengths.
Okay, long waves.
These longer waves are highly effective at penetrating deep water, and more importantly, they resonate violently when they hit a pocket of gas. So when oceanographers use an 18 or 38 kilohertz ping, they are perfectly targeting the gas-filled swim bladders of mesopelagic fishes, like the lanternfish, as well as the pneumatophores, the gas floats of certain siphonophores.
The sound wave hits that tiny internal gas bubble, causes it to resonate, and returns a very strong, distinct acoustic echo. So pinging a fish with a swim bladder is essentially like striking a hollow drum. It echoes loudly.
That's a great analogy. However, if you want to detect the smaller, denser organisms, the crustaceans, the swarms of krill, the tiny copepods that lack any internal gas bladders, those low-frequency, long sound waves simply pass right through them without bouncing back. They are acoustically invisible at 38 kilohertz.
So how do you see them?
To see them, you need shorter, higher frequencies. Scientists shift to using 120 kilohertz. The shorter wavelength physically interacts with the dense chitinous exoskeletons of small zooplankton, providing a clear picture of the crustacean swarms.
But there is a catch with higher frequencies, right? If I play a high-pitched whistle, it doesn't carry as far across a field as a low-pitched bass drum. Higher frequency sounds attenuate or lose their energy much faster in water.
You are touching on the primary physical limitation of acoustic oceanography. The 120 kilohertz frequency is excellent for resolving detail, but its rapid acoustic attenuation restricts its effective range to only the upper layers of the mesopelagic zone. The sound literally dies out before it reaches the bottom of the DSL.
Wow, so you can only see the top half of the swarm.
Exactly.
Right.
And if you want to get even more granular, say, you want to measure microzooplankton, you have to use an extremely high 200 kilohertz frequency. But a 200 kilohertz signal is so delicate and so easily scrambled by the physical turbulence and noise generated by the hull of a moving ship that you can't just mount the sensor on the bottom of a boat.
What do you do instead?
You have to physically deploy the transducer down into the water column via tethered remotely operated vehicles, or ROVs, placing the instrument right in the middle of the biological layer.
So researchers basically had to build a composite image. By blending all these different acoustic frequencies together, the bass, the mid-range, the treble, and integrating that massive data set with advanced ecosystem models generated by global circumnavigation trips like the Malaspina expedition, they completely rewrote the mathematical formulas.
The change in the numbers was staggering.
The old net-based estimate was 1 billion metric tons. The new current acoustic consensus, the global biomass of mesopelagic fish alone, is estimated to be between 10 and 15 billion metric tons.
It is an order of magnitude higher.
That mind-blowing revision makes the deep scattering layer the single largest, most densely packed aggregation of vertebrate life on planet Earth.
And the sheer scale of it forces us to ask a vital ecological question. Why should humanity, living our daily lives on dry land, care about a massive wall of tiny glowing fish hiding 500 meters underwater?
Right. Why does it matter to us?
The answer lies in the movement. Because such an unimaginable volume of biomass participates in this synchronized daily vertical migration, the DSL functions as a fundamental, indispensable component of the Earth's climate regulation system. It is the engine of the biological carbon pump.
The carbon pump is such a brilliant concept. Let's break down exactly how this engine works for the listener. It starts at the very surface of the ocean, in the sunlit euphotic zone. Up there, you have phytoplankton, microscopic marine algae.
The base of the entire food web.
Just like plants and trees on land, these phytoplankton utilize dissolved carbon dioxide from the atmosphere to conduct photosynthesis. They literally capture our inorganic greenhouse gas emissions and convert them into solid, organic, physical biomass, their own bodies.
Traditionally, science viewed the biological carbon pump as a slow, passive system. They called it the gravitational pump.
It's a very peaceful image. It's like autumn leaves falling from a tree in slow motion. Dead phytoplankton, cellular detritus, and bits of organic matter clump together into what we call marine snow. And this snow just slowly, passively, drifts down through the water column into the abyssal depths, taking months or even years to reach the bottom.
That passive gravitational sinking absolutely does occur, but the diel vertical migration of the deep scattering layer totally rewrites the efficiency of the entire system. The migration introduces a massive, active, high-speed biological transport mechanism. Ecologists refer to it as the mesopelagic migrant pump.
If the passive gravitational pump is like autumn leaves drifting on the wind, the migrant pump is a high-speed industrial freight elevator. Every single evening, these billion upon billions of tiny fish, squids, and zooplankton ride the elevator up to the surface layer. Under the cover of darkness, they gorge themselves on the carbon-rich phytoplankton.
They eat as much as they possibly can.
Then, just before the sun rises, their internal clocks trigger, and they ride the elevator all the way back down into the dark, freezing depths, hiding from visual predators to safely digest their massive meal.
And it is what happens at depth that regulates our climate. While resting in the cold, high-pressure mesopelagic zone, these organisms breathe. They undergo respiration, converting oxygen and releasing dissolved organic carbon into the deep water.
So they exhale carbon down there.
Furthermore, and perhaps most importantly for sequestration, they digest their food and excrete solid, highly dense fecal pellets. Because these pellets are dense and compact, they sink incredibly fast, bypassing the slower degradation processes of the upper ocean.
Through this synchronized daily routine of eating at the surface and excreting in the deep, these migrating organisms are actively injecting massive quantities of captured surface carbon directly into the ocean's interior.
Yes.
I want to make sure I'm visualizing this correctly. They are literally swimming up to the surface, eating our atmospheric greenhouse gases, and then swimming down to poop those greenhouse gases into the abyss.
Put bluntly, yes. That is the fundamental mechanism. And the depth is crucial. When this exported carbon is finally remineralized, broken back down by bacteria, it occurs below what oceanographers call the permanent thermocline.
What does that mean for the carbon?
The thermocline is a deep boundary layer of water that does not easily mix with the surface. Once carbon is pushed below the thermocline, it is effectively locked away, sequestered from any atmospheric exchange for centuries, if not millennia.
It's basically trapped in a deep sea vault.
Exactly. Recent biogeochemical models demonstrate that this active biological transport accounts for a highly significant percentage of the total downward carbon flux globally. It is especially vital in oligotrophic ocean gyres.
To clarify for everyone, an oligotrophic gyre is essentially a massive, swirling oceanic desert. Areas far out in the middle of the Pacific or Atlantic where nutrients are incredibly scarce, the water is crystal clear, and not much surface life exists.
In those vast marine deserts, the active transport by migrating twilight zone creatures is sometimes the primary mechanism for pulling carbon out of the atmosphere. The implications are profound.
What happens if that system breaks?
If human activities disrupt the deep scattering layer, for example, if we authorize commercial fishing fleets to drag nets through the mesopelagic to harvest krill and lanternfish to grind into cheap fish meal for agriculture, or if climate-driven ocean warming leads to severe deoxygenation in the midwater, we risk severely breaking the ocean's capacity to buffer global greenhouse gas emissions.
The interconnectedness is just staggering. We rely on microscopic glowing fish to keep the planet from overheating, but it brings up a really interesting follow-up question.
What's that?
If the twilight zone is packed shoulder to shoulder with 15 billion tons of tiny migrating creatures, what happens to the animals that don't commute? What happens to the creatures that are born, live, and die permanently stationed in the frigid, crushing depths of the abyssal plain far below the twilight zone?
Well, nature has evolved a vastly different survival strategy for them. They don't stay tiny, they become absolute giants.
You are describing the architecture of biological extremes, a phenomenon formerly known as deep sea and polar gigantism. It is arguably one of the most visually striking and initially perplexing phenomena in marine biology.
It really is. Organisms forced to adapt to these extreme abiotic constraints. The crushing hydrostatic pressure, the near freezing temperatures, the absolute lack of solar energy responds by undergoing radical morphological transformations that completely defy our understanding of terrestrial scale.
It is genuinely straight out of a science fiction horror movie. To understand how weird this is, let's list some of these monsters of the deep and compare them to what we know on land. Take the common pillbug or the little roly-poly you might find hiding under a wet rock in your garden.
Everyone knows a pillbug.
The deep-sea equivalent of a pillbug is an isopod. In January 2025, scientists conducting deep-water surveys discovered a brand new species of supergent isopod off the coast of Vietnam. It looked so intimidating, they literally named it the Darth Vader isopod Bathynomus vedar.
Which is a fantastic name, honestly.
It is, and this creature consistently grows to over 12 inches in length. Think about a pillbug the size of a loaf of bread. That is 16 times larger than its terrestrial cousin.
And the crustacean gigantism doesn't stop with isopods. Consider amphipods, which on a typical beach are known as tiny, jumping sand fleas barely a few millimeters long.
Like the ones that bite your ankles at the beach.
Exactly those. But when you send robotic landers down into mid-ocean trenches, 7 kilometers beneath the surface where the pressure is incomprehensible, you find scavenging amphipods exceeding 30 centimeters in length. That's over 10 times the size of coastal variants.
It's just wild to think about. What about other animals?
If we look at the phylum Porifera, the sponges' deep-sea expeditions routinely encounter abyssal glass sponges the size of minivans. Because their metabolic rates are slowed to an almost imperceptible crawl by the freezing temperatures, their growth rate suggests these massive sponges may be continuously living for thousands of years.
But as impressive as a minivan-sized sponge is, the absolute undisputed king of the deep-sea giants has to be the colossal squid. And to be clear, I am not talking about the famous giant squid. The colossal squid is a different beast entirely. It lives primarily in the freezing abyssal waters of the southern ocean surrounding Antarctica.
It currently holds the title of the heaviest invertebrate on planet Earth.
We are talking about an animal that reaches 10 to 12 meters in length, up to 40 feet. And it has an estimated weight of 500 to 750 kilograms. That is well over 1,600 pounds of muscle, tentacles, and beak.
It is a true leviathan.
Furthermore, to hunt toothfish in the absolute pitch-black darkness of the bathypelagic zone, it has evolved eyes the size of large dinner plates. They're the largest eyes documented in the entire animal kingdom.
The existence of these massive creatures has fueled a fierce scientific debate for decades. The core question is, why? What are the physiological and evolutionary drivers that actively permit or perhaps mandate such massive biological growth in environments that are so hostile to life.
Right, because it doesn't intuitively make sense.
Exactly. Historically, the leading explanation accepted by biologists was known as the oxygen temperature hypothesis, which was first articulated by a biologist named von Bertalanffy back in 1960.
Okay, let me try to walk through the logic of this 1960s theory because I want to explain it and then I want to immediately push back on it because honestly it has never made total sense to me.
Go for it.
The classical theory goes like this. The maximum body size of any cold-blooded marine animal is strictly limited by the physical ratio of its oxygen supply to its metabolic demand. If the water gets warm, the animal's internal metabolism naturally speeds up, like a revving engine, meaning it needs way more oxygen to survive.
That's the baseline, yes.
However, warm water physically cannot hold as much dissolved oxygen gas. So the animal hits a physical wall where it can't absorb enough oxygen to grow larger. Its growth is stunted.
Conversely, in the deep sea, the water is freezing cold consistently between 0 and 4 degrees Celsius.
So the animal's metabolic engine slows down to an absolute crawl. Meanwhile, freezing cold water acts like an incredible sponge for gases, holding a massive amount of dissolved oxygen. So the 1960 theory concluded that this massive abundance of available oxygen, combined with almost zero metabolic demand, theoretically releases the animal from the physical limits of oxygen diffusion.
Without that barrier, their cells and bodies can just expand and expand and expand indefinitely. That mechanism, the release from oxygen limitation, was the accepted, unchallenged paradigm in marine biology for over half a century.
But here's my major issue with it. If a deep-sea creature has a metabolic rate near zero because the water is so freezing, wouldn't carrying around a giant massive body be a huge evolutionary liability?
In what sense?
The deep sea is famously a nutritional desert. There is zero sunlight, meaning there are no plants or algae to eat. The entire ecosystem relies almost exclusively on that slow, meager trickle of marine snow we just talked about, or the incredibly rare lottery ticket event of a dead whale carcass falling from the surface.
Which doesn't happen often.
Exactly. How on earth do you sustain the caloric needs of a 12-inch roly-poly, let alone a 1,600-pound squid, in an absolute desert where there is basically no food?
Your skepticism is incredibly astute, and it mirrors exactly the direction modern ecology has taken over the last decade. Researchers began to realize that physiological limits alone don't explain the active advantage of gigantism.
So what's the newer theory?
Other theories started gaining traction, theories that heavily emphasized resource dynamics and ecological strategy over just oxygen physics. It fundamentally comes down to an ecological principle known as Bergmann's rule, specifically as it is applied to the deep sea. The key concept to understand here is mass-specific metabolic rates.
Let's define that. What exactly does a mass-specific metabolic rate mean for an animal?
It means that as a body gets larger, its overall metabolism might increase, but the amount of energy required per gram of tissue actually decreases. Larger bodies have significantly slower mass-specific metabolic rates.
So they are more efficient per pound.
Crucially, they also possess vastly greater volumetric capacities for energy storage. Massive bodies can store vast, dense reserves of lipids. They can essentially pack on incredible amounts of fat.
Like a camel storing fat for the desert.
Exactly like that. This provides an incredible degree of metabolic efficiency, allowing deep-sea giants to endure remarkably prolonged periods of starvation when food is scarce. To give you an extreme example, giant isopods captured and kept in laboratory captivity have been scientifically documented surviving for over 5 full years without consuming a single meal.
5 years without eating. That is metabolic optimization taken to the absolute extreme. So the size isn't a liability, it's a giant walking battery.
And there's a second major factor. Because they are so isolated, spread out across the massive abyssal plains, there's an extremely low density of apex predators.
Which changes the evolutionary math completely. On a terrestrial savanna or in a shallow sunlit coral reef, if you are big, slow, and full of fat, you quickly become someone else's lunch. You get eaten.
Right. But in the deep sea, you don't have that relentless evolutionary pressure forcing you to stay small, hide in cracks, and breed quickly. You can take your time, drastically delay your sexual maturity, and allocate almost all of your biological energy into prolonged, continuous physical growth.
The ecological strategy is to become an unassailable storage tank. I love that.
Yeah.
And the true final paradigm shift regarding the old 1960 oxygen temperature hypothesis occurred very recently, right?
It did. In 2019 and 2020, researchers decided to finally conduct a rigorous series of laboratory experiments on giant Antarctic sea spiders technically known as pycnogonids, specifically studying species from the genera Culissendice and Amathea.
What were they trying to prove?
Their goal was to physically stress test the 60-year-old von Bertalanffy hypothesis.
Right, because the logic dictates that if physical oxygen diffusion through their skin is the only thing keeping them alive at that massive size, then if you artificially lower the oxygen levels in a tank, the absolute biggest spiders should suffocate and die first. Their mass would be too huge relative to their surface area to absorb enough dwindling oxygen.
That was the foundational expectation of the experiment. The researchers placed these giant sea spiders into controlled chambers and subjected them to varying levels of dissolved oxygen, a state called hypoxia, while simultaneously elevating the water temperatures to increase their metabolic demand.
Okay, pushing them to the brink. What happened?
The empirical results were shocking. The data showed no significant interaction whatsoever between the physical body size of the spider and its vulnerability to oxygen limitation. The giant, dinner plate-sized sea spiders were not inherently more vulnerable to hypoxia than the tiny, thumbnail-sized ones.
Which completely shatters the 1960 rule. If the surface area physics say they shouldn't be able to breathe, how are the massive spiders surviving the low oxygen?
The researchers had to look at the spiders under scanning electron microscopes to figure it out. The morphological analyses revealed an incredible, entirely unexpected physiological adaptation. As these pycnogonids grow larger and age, their cuticles, their rigid outer exoskeletons, do not remain static.
What happens to the shell?
The exoskeleton becomes increasingly mechanically porous. It fills with microscopic holes. The animal physically alters its own surface architecture as it grows to mechanically compensate for the longer diffusion distances inside its body. By creating a sponge-like exoskeleton, it vastly increases the physical pathways for oxygen to reach its internal tissues.
That is evolutionary genius.
Yeah.
They literally drill holes in their own armor to breathe better. So when we synthesize all of this, what is the modern consensus on the science of gigantism?
The modern biological consensus has profoundly shifted. We now understand that freezing oxygen-rich environments are strictly a permissive factor, meaning, yes, deep-sea gigantism probably cannot initiate without those specific physical conditions being present.
But they are not the evolutionary engine driving the change.
Exactly. The true driver is ecological necessity. The absolute requirement for metabolic optimization, the desperate need to store massive energy reserves in a food-scarce biological desert, and the evolutionary release provided by low predation pressures.
The deep sea forces you to become a hyper-efficient giant storage tank just to survive the famine. It's a brilliant strategy. But, listening to this brings up the next massive logical puzzle.
Let's hear it.
If these animals are so evasive, or if they live thousands of meters down in the bathypelagic zone, where the crushing pressure instantly implodes our standard cameras and equipment, how on earth do we actually study them? How do scientists track massive, terrifying squids or inventory the billions of delicate jelly animals in the twilight zone without just blindly dragging a destructive net through the dark and hoping for the best?
To solve that puzzle, marine biologists had to stop looking for the animals themselves and start looking for the invisible traces they leave behind. We are talking about the revolutionary science of environmental DNA, commonly referred to as eDNA.
The genetic dust.
Yes. The implementation of this technology has utterly and completely revolutionized marine exploration over the past decade. For over a century, as we discussed with the nets, marine biology relied heavily on extractive physical sampling. Trawls, benthic dredges, sediment sleds.
All the destructive stuff.
It's highly destructive, it's severely biased against fast-swimming or fragile organisms, and practically speaking, it is vastly inadequate for sampling the massive three-dimensional volume of the global ocean. Environmental DNA allows scientists to map entire webs of biodiversity simply by extracting and analyzing a single bottle of seawater.
Because everything living in the ocean is constantly shedding parts of itself into the water. It's a bit gross when you really think about it, but every fish, every crab, every squid, and every whale is constantly leaving a microscopic trail of genetic dust wherever it goes.
They shed feces, they shed metabolic waste products, they're constantly sleeving off dead epidermal skin cells and loose scales.
And mucus. So much mucus.
They secrete massive amounts of mucus, yes. Even when a massive cetacean, like a sperm whale, breaches the surface and forcefully exhales through its blowhole, it deposits a highly concentrated mixture of respiratory tract cells and lung mucus directly into the surface water.
All of this biological detritus, this genetic dust, drifts through the water column, often becoming incorporated into the sinking marine snow. It creates a vast, invisible, suspended genetic footprint of the entire surrounding ecosystem.
To harness this invisible data, researchers collect liters of raw seawater, often using automated rosette samplers deployed from ships, and they physically pass that water through incredibly fine microfilters. The filters capture all the suspended cellular material.
Then what do they do with the filters?
The researchers take those loaded filters back to a sterile laboratory, chemically extract the raw DNA from the cells, and subject it to one of two distinct, highly specialized bioinformatic methodologies.
Okay, let's break these two methodologies down clearly because understanding the difference between them is crucial to understanding how we track ocean life. The first major method is called targeted detection, which uses laboratory techniques like UPCR or ddPCR. This is a tool researchers reach for when they are looking for one very specific, already known organism.
Yes, targeted detection is highly specific. The process involves using short, artificially synthesized sequences of DNA, which scientists call primers. These primers are engineered to act like highly selective molecular magnets.
How does it pick the right DNA out of the water?
When you process a water sample, you are dealing with a massive alphabet soup of environmental DNA floating around, billions of fragments from thousands of different creatures. The specific primers are designed to completely ignore all the background noise of the other organisms. They will mathematically bind only to the complementary DNA sequence of the designated target species.
Once bound, laboratory enzymes can exponentially amplify that specific DNA fragment until it is easily quantifiable by a computer.
Exactly.
I always like to visualize targeted detection like a bouncer working the door at an incredibly exclusive club with a strict VIP guest list. The bouncer only has one single name written on his clipboard. Let's say the target is a specific, highly endangered species of sea turtle.
I like this analogy.
The bouncer can scan a massive crowd of 1,000 random people, completely ignore 999 of them, grab the turtle, and definitively say, aha, you were in this room. It is exceptionally sensitive. In fact, field scientists utilize this exact targeted method to monitor extremely evasive or rare megafauna that they can never seem to catch on camera.
They've even used it in freshwater rivers and deep lakes to rediscover amphibian species that classical biology had declared entirely extinct. It is an incredibly powerful forensic tool for known targets.
But what happens when your scientific goal isn't to track down one specific known animal? What if your mission is to conduct a comprehensive baseline biodiversity assessment of an entire coral reef ecosystem?
Or, more challenging, what if you were trying to discover entirely unknown ecosystems hidden in the abyssal plains? That is where the second methodology becomes vital: metabarcoding.
If targeted detection is the VIP bouncer looking for one name, metabarcoding is like the police locking the doors to the entire club, forcefully taking the fingerprint of every single individual person inside, and then running all of those fingerprints through a massive international Interpol database at the exact same time to see exactly who is in the building.
That is a highly accurate analogy. Instead of synthesizing a primer for a single species, scientists utilize what are called universal primers. These universal primers are designed to target highly conserved genetic loci.
What does that mean practically?
It means they target specific regions of DNA that have remained relatively unchanged across massive evolutionary timescales, meaning they are shared across incredibly broad taxonomic groups.
Give me an example.
For example, a geneticist might use a primer targeting the mitochondrial cytochrome C oxidase subunit I gene, commonly known as the COI gene. The COI gene is incredibly effective for identifying mazoans, which basically encompasses all complex animal life.
Okay, so that catches all the animals.
Alternatively, if they want to inventory the microscopic base of the food web, they might use universal primers targeting the 16S and 18S RNA genes, which amplify the DNA of vast arrays of bacteria and broader eukaryotes. By using these universal primers, a single sequencing run can simultaneously amplify the DNA fragments of hundreds or even thousands of different species from one single bottle of water.
So you run the machine, and it spits out this impossibly complex mixture of thousands of different genetic sequences. You then take that massive digital file and cross-reference it against global public reference databases, massive digital libraries like GenBank, or the Barcode of Life data system.
The computer rapidly matches the sequences and generates a complete, itemized inventory of every organism that left its genetic traces in that water.
It genuinely sounds like science fiction magic.
Yeah.
But I know ecologies is never that simple. There must be severe limitations, because the deep ocean isn't a sterile, static laboratory beaker.
You've identified the core challenge. The ocean is a highly dynamic, endlessly churning fluid environment. This physical reality introduces the complex science of taphonomy.
Taphonomy.
Yes. Taphonomy is the study of the physical, chemical, and biological processes that govern the degradation, the physical transport, and the ultimate environmental fate of a DNA molecule after it is shed by an organism.
So what happens to the DNA over time?
Exactly. Because merely detecting DNA is not enough. Extracting accurate, actionable ecological meaning from an eDNA hit requires deeply understanding how long that fragile molecule physically survives in the water column before it disintegrates.
And from what I understand, that survival time varies wildly depending on the environment, specifically based on the ambient temperature and the amount of sunlight hitting the water, right?
Parastically, it is a completely different taphonomic environment depending on where you are. In shallow, warm, sunlit coastal waters, say a tropical coral reef, you have incredibly high microbial and enzymatic activity. The water is full of bacteria aggressively breaking down organic matter.
Plus, the sun is beating down on it.
Right. You have intense ultraviolet radiation from the sun penetrating the shallows, which physically shears and destroys DNA strands. Because of these harsh factors, eDNA in surface waters degrades incredibly rapidly. Studies show it often drops below the limits of scientific detection within just a few hours, or at most, a few days.
But when you take your robotic sampler down into the deep ocean, to the bathypelagic or abyssal zones, it's a completely different preservation story.
Exactly. Recent laboratory experiments designed to precisely simulate deep-sea conditions revealed what scientists call a biphasic decay model for marine eDNA. The initial phase of degradation is relatively rapid, no matter the environment, as the weakest cellular membranes rupture.
Okay, what's the second phase?
The secondary long-term decay phase is strongly, almost entirely controlled by the ambient water temperature. In the absolute dark, extreme high-pressure, freezing environments of the deep sea, specifically at temperatures at or below 10 degrees Celsius, the enzymatic breakdown of DNA almost halts entirely.
So it basically goes into cold storage.
Yes. Experimental data definitively show that marine eDNA can persist at highly quantifiable concentrations in the deep sea for over 2 full weeks. Compare that to water at 20 degrees Celsius or warmer, where the genetic signal degrades completely into static in a week or less.
So a genetic footprint laid down in the deep sea lasts more than twice as long as one on a sunny beach. But structurally, that extended lifespan actually creates an entirely different forensic problem for biologists. The ocean is not a frozen block of ice. It is constantly moving.
It is a fluid, yes.
If you pull up a water sample from the abyssal plain and the computer says you found the eDNA of a bizarre deep-sea anglerfish, it doesn't necessarily mean that anglerfish is sitting right there staring back at your robotic camera.
Correct. The fluid dynamics of the ocean complicate everything. You have massive, slow-moving, deep ocean currents which cause a process called advection, the horizontal transport of the water mass itself.
Yeah.
And you have turbulent mixing, which causes diffusion, the spreading out of the genetic particles.
So the DNA could have drifted for miles.
These physical forces continuously transport eDNA miles away from its biological point of origin. Furthermore, when scientists conduct deep-sea benthic surveys, taking sediment cores directly from the ocean floor, they have to constantly account for the problem of vertical contamination.
Oh, this goes right back to the marine snow we discussed with the carbon bomb.
Exactly the same mechanism. DNA that is shed by pelagic fishes or squids swimming thousands of meters above in the sunlit twilight zone eventually binds to detritus, sinks down as marine snow, and accumulates in the abyssal mud.
So you might sequence mud from the deep sea floor and find tuna DNA, even though a tuna has never swum that deep. To accurately pinpoint the spatial and temporal origin of a biological signal, to definitively answer whether an animal was physically present 1 mile away yesterday, or 10 miles away a week ago, or a mile above you a month ago. Scientists cannot simply read the raw DNA output.
They are forced to integrate that biological eDNA data with highly complex, supercomputer-driven hydrodynamic ocean drift models.
It is quite literally forensic oceanography. You have to calculate the current, the decay rate, and the genetic match to solve the crime of who was there. But here is where we hit the most glaring, fundamental flaw in the entire metabar coding system. Let's go back to our club analogy. You lock the doors, you take the fingerprints of every single organism in the water, you run all of them through the massive GenBank database. What happens when the computer database comes up completely empty?
That is the big question.
What happens when you scan a genetic barcode from the abyss and the computer just flashes unknown?
This specific limitation brings us to the most daunting challenge in modern marine biology, the dark taxa conundrum. When researchers filter water or scoop sediment from the abyssal plains and sequence the resulting eDNA, they frequently encounter an astonishing, almost discouraging phenomenon.
How bad is it?
In some deep ocean samples, up to 90% of the successfully recovered genetic sequences do not match anything currently cataloged in the global reference repositories.
90%. So we had the genetic fingerprints of thousands of creatures, but absolutely no idea what they look like or what they do.
Let's contextualize the sheer scale of this problem by looking at a highly specific, heavily scrutinized region. In 2023, marine biologists conducted a massive, unprecedented biodiversity synthesis of an area known as the Clarion-Clipperton Zone, or the CCZ.
Okay, where is the CCZ?
The CCZ is a staggering 6 million square kilometer expanse of the abyssal Pacific Ocean, stretching roughly between Hawaii and Mexico. It is an area of intense geopolitical focus because it is heavily targeted by international conglomerates for deep-sea mining, which we will explore in depth shortly.
Right.
To establish a scientific baseline of exactly what ecological wealth might be destroyed by mining, researchers systematically compiled over 100,000 individual biological records from 7 primary deep-sea data sources to catalog the benthic metazoan biodiversity, the complex animal life living on the seafloor in that specific zone.
And the findings from that synthesis were absolutely staggering. After crunching all that historical and genetic data, they recorded a total of 5,578 distinct animal species living in the CCZ. But an estimated 88% to 92% of those species, we're talking about 5,142 distinct living organisms, were entirely unnamed and completely new to science.
They currently exist in our records only as informal, blurry morphological photographs or as isolated, orphaned genetic sequences in a computer file.
The pervasive presence of these dark taxa stems from 2 distinct scientific bottlenecks, right?
It does. First, an organism might actually be known to classical taxonomy. It might be one of those specimens sitting in a museum jar in Paris or London, possessing a formal Latin name, but because it was collected decades ago, no one has ever extracted and sequenced its genome.
So it remains completely invisible to modern, automated eDNA matching databases.
Exactly. The animal has a face and a name tag on a jar, but the police don't have its genetic fingerprint on file. So when the DNA shows up in the water, the computer can't connect the two.
What's the second bottleneck?
The second bottleneck, which is statistically far more common in the extreme deep sea, is that the organism shedding the DNA is an entirely novel, undiscovered species that represents a totally unknown branch on the evolutionary tree of life.
It has no face, no name, no physical specimen in a jar, and no closely related genetic cousins in the database. And as we discuss heavily with the taxonomic impediment, classical taxonomy takes an average of 13.5 years per single species. We simply do not have the human lifespan or the scientific funding to resolve 5,000 unknown species the old-fashioned way.
This is exactly where Silicon Valley engineering is meeting the Mariana Trench. Because human biologists can't keep up, we are bringing in artificial intelligence. Traditional bioinformatics relies heavily on strict alignment-based sequence matching.
Meaning you compare 2 strings of letters.
Right. You have sequence A from the ocean, you look for a sequence B in the database that is 99% identical, and you declare a match. But that simplistic system completely fails when the organism is so novel that it has no closely related reference sequence. AI, however, is being trained to bypass this limitation entirely.
It is a brilliant application of machine learning. Instead of looking for a direct one-to-one matching copy, the AI is trained to recognize the underlying, highly complex mathematical and structural patterns hidden within the raw genomic architecture itself.
Scientists are increasingly deploying advanced AI to decode dark taxa. For example, there is a powerful algorithmic tool called Bayesian. It functions as a Bayesian non-parametric taxonomic classifier.
That's a mouthful.
It is. But it was engineered specifically to predict the taxonomic affiliation of entirely unknown, deeply orphaned DNA sequences.
I am fascinated by how a computer algorithm can actually do biology. How does it look at a string of ATCG letters and know what kind of animal it came from without a reference? Does it translate the DNA into a different language?
In a highly complex mathematical way, yes. Advanced models, specifically deep neural networks, or DNNs, and convolutional neural networks, CNNs, are utilized to map the raw input sequence data. They break the DNA down into smaller mathematical fragments known as chimeras.
Cameras, okay.
Some of the most cutting-edge algorithms actually mathematically translate pairs of DNA bases into complex, multidimensional images that represent the subtle physiochemical properties and structural folding tendencies of the molecule.
Wait, let me make sure I heard that correctly. The computer is literally turning a strand of genetic DNA into an abstract picture.
Essentially, yes. It translates genetic code into a structural topology. By analyzing these abstract representations, the machine learning models can identify deep, non-obvious structural and compositional similarities.
So the model can analyze a piece of completely orphaned dark taxa DNA from the CCZ, and the AI can confidently predict its phylogenetic placement. The computer might effectively deduce, I do not have a direct match for this sequence, and I don't know its name, but based on the unique mathematical architecture and K-mer distribution, it is mathematically highly probable that this DNA belongs to a totally undiscovered genus of deep-sea glass squid, or perhaps a novel family of benthic polycatron worms.
That is absolutely mind-blowing. It allows researchers to rapidly cluster these millions of unknown dark taxa sequences into functional ecological groups without ever needing to successfully catch the physical animal in a net.
You can generate vital, predictive biodiversity heat maps for threatened, politically sensitive regions like the CCZ in a fraction of the time. But even with the most powerful AI in the world, the algorithms still need better foundational training data. A neural network is only as smart as the data it trains on.
Which is exactly why we are seeing the launch of these massive, multi-billion-dollar global genomic initiatives racing to physically sequence the ocean's biodiversity.
Absolutely. The global scientific community recognized this bottleneck and has aggressively mobilized to construct the comprehensive, high-fidelity reference databases necessary to fully leverage eDNA and AI. They refer to this massive shift as a transition to high-throughput cyber taxonomy.
Perhaps the most ambitious and staggering of these initiatives is the Earth Biogenome Project, or EBP. The leading scientists and founders of the project literally describe it as biology's equivalent to the Apollo moonshot.
And their stated goal is wonderfully, terrifyingly wild. Over a tight 10-year period, they want to physically sequence, painstakingly catalog, and fully characterize the high-quality reference genomes of all 1.67 million named eukaryotic species on planet Earth. Every single complex organism we know exists.
They have already successfully completed Phase 1, which focused on establishing the complex technological pipelines, and their Phase 2 goals are phenomenally aggressive. Over the next 4 years alone, the EBP aims to generate complete reference genomes for 150,000 species.
Hitting that target ensures that at least 50% of all currently accepted eukaryotic genera are officially represented in a permanent digital library of life.
To maintain that punishing timeline, the network of global sequencing centers has to successfully assemble, verify, and publicly release more than 3,000 brand new genomes every single month. The budget allocated for this specific phase alone is estimated at a massive $1.1 billion.
It really is the Manhattan Project for biological cataloging. And running completely parallel to the Biogenome Project, you have initiatives like the Ocean Census, which was launched recently in 2023 by the Nippon Foundation and the Necton Marine Institute. The Ocean Census teams are the ones actively doing the physical cyber taxonomy out on the water.
They collect the specimens and immediately merge rapid DNA sequencing with high-resolution 3D digital imaging to create comprehensive, interactive digital twins of marine organisms.
And crucially, rather than hoarding the data and siloing it in expensive, paywalled academic journals for 13 years while they wait for formal publication, they actively push all of their raw discoveries directly to open access, public platforms like OB, the Ocean Biodiversity Information System, and WORMS.
That open data, open source architecture is the defining shift in modern conservation. It immediately transforms hidden academic biodiversity into actionable, legal data that can inform international conservation policy before the formal taxonomic publication is ever printed.
We also must acknowledge the incredible historical foundation laid by the Terra Oceans expedition.
Yes, the Terra Oceans expedition was a monumental, multi-year transoceanic survey. They sailed around the world, collecting roughly 40,000 plankton and microscopic samples globally. They generated a mind-bending 250 billion raw DNA sequences.
During their analysis, they uncovered over 100,000 entirely new species of single-celled microalgae, and they mathematically multiplied the known types of marine viral DNA by more than 100-fold. They essentially cataloged a database of 40 million novel microbial genes. They single-handedly sequenced the invisible, foundational base of the entire global food web.
But while microscopic dark taxa and single-celled organisms represent the overwhelming numerical bulk of unknown marine biodiversity, the deep ocean continues to conceal absolute, massive giants from our cameras. We have to talk about how eDNA is specifically helping us hunt the ocean's greatest ghost, the colossal squid.
Right. I asked earlier, and I've been waiting to get back to this, how do we actually know they get to be 1,600 kilos and 40 feet long if they live so deep?
Yeah.
If we've never properly studied a live, healthy adult in its natural abyssal habitat, where do biologists get those highly specific numbers?
It is a great question, and the answer highlights how patchy our historical knowledge is. Our current biological understanding of the colossal squid comes almost entirely from two rather gruesome opportunistic sources.
Which are?
The first source is dead or rapidly dying specimens that become entangled and hauled to the surface as accidental bycatch by commercial deep-sea toothfish longliners. The second, more prolific source is the indigestible, razor-sharp chitinous beaks extracted from the stomachs of dead, air-breathing sperm whales.
Sperm whales are known to dive thousands of meters into the freezing abyss, specifically to hunt these massive squids in epic, unseen battles. It's incredible to think that the largest, heaviest invertebrate on the entire planet is essentially a biological ghost. We only know it exists because a whale ate it or a fisherman accidentally snagged it.
It is incredibly frustrating for biologists.
Why can't we just use some of that $1.1 billion budget, send a high-tech submarine down to the Southern Ocean and just film them swimming around?
Because the standard technology we use actively repels them. Standard ROVs are massive, clunky machines. They emit intense, blinding artificial lights to pierce the dark, and their mechanical thrusters generate a massive amount of acoustic noise.
And these animals are adapted to silence and darkness.
Exactly. Animals like the colossal squid, which have evolved eyes the size of dinner plates specifically adapted to the absolute pristine darkness and near silence of the bathypelagic zone, are instantly blinded and terrified by the ROV. They use their massive jet propulsion to flee the area long before the camera's limited field of view can ever capture them.
So you can't kick the front door down. You need extreme stealth.
Exactly. Deep-sea videography of megafauna requires specialized passive stealth technology. This specific approach actually yielded massive headline-making success back in 2012. Researchers utilized specialized red LED lights. Red light occupies a wavelength that is quickly absorbed by water and is completely invisible to the visual spectrum of most deep-sea creatures.
So the animals can't even see the lights.
Right. They combine these invisible lights with passive, bioluminescent mimicking optical lures and successfully captured the first ever high-definition footage of the giant squid, Architeuthis dux, calmly swimming in its natural habitat off Japan.
And now, researchers are actively turning that exact same stealth technology toward the freezing waters of the colossal squid. There is a fantastic nonprofit organization called Colossal that is partnering directly with commercial polar tourism vessels. They're dropping these modified stealth camera systems into the deep trenches around the Antarctic Peninsula.
And we actually have incredible breaking news on this front. Just recently, in March 2025, in a monumental breakthrough for marine biology, the Schmidt Ocean Institute captured the very first ever in situ video footage of a juvenile colossal squid in the wild.
It was a spectacular achievement. The footage was recorded at a depth of roughly 600 meters near the remote South Sandwich Islands in the South Atlantic. The juvenile they filmed was only about 30 centimeters long. Interestingly, it prominently displayed the transparent, almost glass-like physiology that is completely typical of its broader taxonomic family, the Cranchidae, often called glass squids, before they mature and develop their thick, massive red musculature.
A 30-centimeter transparent baby that will eventually grow into a 10-meter, 1,600-pound titan. And to find the adult hunting grounds, researchers aren't just blindly dropping cameras into the massive Southern Ocean anymore.
They are utilizing eDNA. They collect water samples at various depths and latitudes across the Antarctic, and they utilize those highly targeted qPCR primers we discussed earlier, the VIP bouncer list, specifically coded to detect cephalopod DNA.
By mapping where the DNA hits are strongest, they are building highly accurate genetic heat maps to identify the colossal squid's deep water spawning and hunting grounds without ever needing to lay eyes on the actual animal.
And beyond the sheer awe of the squid, the abyss is routinely yielding other monster discoveries every single year. We talked about the massive Darth Vader isopods found off Vietnam. There are gigantic carnivorous death ball sponges actively feeding in the Southern Ocean. There are shimmering glacial glass squids being revealed in the newly exposed waters beneath collapsing Antarctic ice shelves.
The deep ocean is absolutely not a barren, empty wasteland. It is a highly dynamic, fiercely competitive ecosystem hiding giants just beyond the reach of our submarine lights.
But right here is where we have to make a tragic, urgent pivot. The frantic rush to map these dark taxa, the multi-million-dollar race to finally film the colossal squid, the massive sequencing projects. It isn't just driven by pure, innocent academic curiosity.
Unfortunately, no.
We're standing on the precipice of aggressively industrializing the deep ocean before we even possess a basic understanding of how it functions.
The scientific urgency is directly driven by a rapidly closing, very narrow window of opportunity. The deep sea is currently facing the imminent existential threat of global deep sea mining. And the complex irony here is that this drive to industrialize the abyss is largely propelled by the global political and economic transition toward green energy.
Specifically, the voracious exponential demand for transition battery metals. It is an incredibly painful, almost cruel irony. In our desperate attempt to save the terrestrial climate and transition away from fossil fuels, we are actively looking at destroying the largest intact ecosystem on Earth to build the batteries.
And the absolute epicenter of this looming environmental crisis is the exact area we talked about earlier. The CCZ, the Claring-Clipperton Zone in the Abyssal Pacific. The muddy plains down there are literally carpeted with things called polymetallic nodules.
To understand the mining appeal, you have to understand the nodules. These polymetallic nodules are roughly the size and shape of potatoes. They sit loosely on the abyssal mud, but they are geological marvels. They take millions upon millions of years to naturally accrete layer by microscopic layer, precipitating metals out of the seawater.
Because of this slow process, they are highly enriched in extremely valuable industrial metals: cobalt, nickel, copper, lithium, and rare earth elements. Several international mining conglomerates, most notably a corporation called the Metals Company, operating via its subsidiary Nauru Ocean Resources, view these vast deep-sea reserves as the ultimate necessary solution to impending terrestrial mineral shortages.
They are currently aggressively lobbying the International Seabed Authority, the UN body known as the ISA, to transition their current scientific exploration permits into full-scale commercial extraction licenses.
What actually happens to the ocean if the ISA grants those licenses and the commercial machines turn on? The ecological stakes are severe, and from everything I've read in the environmental impact reports, the damage will be completely irreversible on any timescale relevant to human civilization.
The physical mechanical destruction of the seafloor is the most immediate and localized threat. As we established, deep-sea biological communities operate on vastly extended geological timescales. The species living in the CCZ are highly specialized, extremely long-lived, and display incredibly low reproductive rates.
But more fundamentally than just the slow biology, the polymetallic nodules themselves are the vital habitat.
Right, I want to emphasize this, because the abyssal plain is mostly just endless, flat, millions of square miles of soft, gooey sediment. The nodules are the only solid, hard rocks around for miles. For the animals that can't swim, these potato-sized rocks serve as critical, obligate, hard substrates.
Exactly. Sessile organisms, animals permanently anchored in place, such as delicate deep-sea glass sponges, ancient slow-growing corals, and various anemones, cannot survive in soft mud. They anchor themselves directly onto the hard surface of the nodules.
The proposed industrial harvesting process involves massive, heavily weighted, tank-like machines driving relentlessly across the delicate seafloor to physically vacuum up and remove these millions of years old formations. That process results in the permanent, physical eradication of the foundational substrate. It guarantees the absolute localized extinction of all nodule-dependent fauna within the mining tract.
But the devastation doesn't neatly stay localized strictly to the caterpillar tracks of the machines. You have the secondary effect of mechanical dredging, which severely compacts the seabed and generates massive, towering, benthic sediment plumes.
These giant, thick clouds of disturbed, pulverized mud drift unpredictably with the slow, deep ocean currents. As they settle, they bury small organisms living in the sediment, and they completely smother the delicate, highly specialized feeding apparatuses of suspension feeders over vast areas far beyond the actual mining footprint.
And critically, the environmental threat extends vertically, reaching all the way up into the mesopelagic twilight zone we discussed earlier. The proposed mining process involves pumping the collected slurry, Chaotic mixture of mud, crushed organisms, and the nodules up through miles of vertical riser pipes to a massive surface processing vessel.
On the ship, they separate the valuable ore, but then they have to dispose of the massive volume of wastewater and leftover toxic sediment. The current engineering plan is to discharge this waste directly back into the midwater column.
They are going to dump the waste right into the middle of the deep scattering layer, the exact layer that controls the global carbon pump.
Yes. The implications of this were heavily analyzed in a critical 2024 environmental impact study that specifically modeled this midwater discharge. The researchers found that the resulting murky, highly concentrated sediment plumes could physically and physiologically harm up to 53% of the zooplankton and 60% of the micronectin residing in the twilight zone.
By doing what exactly? Is it just clouding the water so they can't see, or is it actually toxic?
It is multifaceted and often much worse than simple toxicity. First, the dense sediment physically blocks and absorbs the bioluminescence that these organisms rely on for interspecies communication, finding mates, and executing their counter-illumination camouflage, effectively blinding them and exposing them to predators.
More insidiously, the suspended sediment drastically dilutes the nutritional value of the naturally occurring marine snow. The twilight zone filter feeders, which are adapted to catch organic particles, inadvertently start consuming the suspended, jagged, inorganic sediment. They're essentially filling their stomachs with completely indigestible, heavy, inorganic junk food.
This threatens to physically starve the foundational base of the pelagic food web. And by extension, starving the migrators threatens to significantly disrupt the efficiency of the biological carbon pump we established earlier.
That is utterly terrifying. You inadvertently starve the fish, and in doing so, you simultaneously break the massive biological carbon pump that is actively buffering our atmosphere from runaway greenhouse gases, all for batteries to save the climate.
And the worst part is, there is zero biological resilience down there. In 2023, scientists involved with the SmartX project went back to revisit a specific deep-sea mining test site in the Pacific Ocean. This specific site was initially dredged 44 years prior, back in 1979, using early, much smaller experimental equipment.
When the 2023 researchers sent their cameras down to look at the 8-meter-wide tracks left by the industrial machines 44 years later, the visual was shocking. The tracks looked like they had been violently carved into the mud yesterday.
The lack of recovery was sobering. The researchers observed extremely stark signs of bacterial or microbial recolonization within the disturbed sediment, but the overall complex animal abundance and species diversity within the machine tracks remained heavily predictably depressed compared to the immediately adjacent untouched areas. The empirical data proves the deep sea simply cannot heal itself on human or even civilizational timescales.
I saw a photo of those tracks, and the best way to describe it is like looking at the famous Apollo astronaut footprints on the moon. They are perfectly preserved because there is no wind or weather to erase them.
Except this isn't a dead, sterile rock floating in space. It's our own living planet. Between the looming threat of industrial mining, the ongoing pressure of climate-driven ocean warming, and the progressive deoxygenation of the water column, we threaten to extinguish thousands of incredibly unique dark taxa before we ever have the technological opportunity to sequence their DNA or understand what biological miracles they actually perform.
Which represents a profound, almost incalculable intellectual and biological loss. Because as our technology improves and we look closer at these threatened extreme zones, we are actively finding novel ecosystems that completely rewrite the fundamental textbook laws of biology and thermodynamics.
I really want to pivot our focus to those specific alien discoveries, because this is where the sheer wonder of the deep sea truly shines. For decades, the absolute scientific orthodoxy held that all complex, multicellular ecosystems on Earth, ultimately somewhere down the food chain, relied on the energy of the sun.
Photosynthesis plants converting sunlight into energy was viewed as the indispensable, mandatory base of the global food web. The rule was simple: no sun, no complex life.
That entire biological paradigm was shattered completely in 1977. That was the year geologists exploring along the mid-ocean ridges, the volcanic seams where tectonic plates pull apart, discovered deep-sea hydrothermal vents.
Scientists peering out of the portholes of the Alvin submersible found incredibly vibrant, densely packed biological communities thriving in total, absolute darkness. At these volcanic fissures, superheated, mineral-rich fluids, often reaching temperatures over 400 degrees Celsius, billow out from the Earth's crust into the freezing ocean.
And the life there operates via an entirely alien mechanism. Chemosynthesis, not photosynthesis. I remember learning about this. You have these specialized extreme bacteria and archaea that can actually oxidize the toxic hydrogen sulfide and other harsh chemicals spewing from the vents. And they use that chemical reaction to produce organic energy.
They replace the plants. They form the base of a wild alien food web that supports bizarre creatures like giant riftia tube worms with these bright blood-red plumes, highly specialized vent grabs, and swarms of blind, pale shrimp. It's a completely alien world right here on Earth.
But recent research expeditions in 2024 and 2025 have vastly expanded our understanding of where these sunless habitats can actually exist.
Yes, the discoveries are accelerating. In 2024, during the Ocean Census Arctic Deep Expedition, an international team made a monumental find. They discovered what are now called the Freya Hydrate Mounds.
This complex ecosystem is located at a staggering depth of 3,640 meters along the Malloy Ridge, deep in the freezing Greenland Sea. This specific discovery represents the deepest known gas hydrate cold seep ever found on Earth, pushing the known depth limit for these ecosystems by nearly 1,800 meters.
Let's clarify the terminology for a moment. How does a cold seep physically differ from the boiling hydrothermal vents we just described?
Unlike hydrothermal vents, which are powered by shallow magma chambers and are literally scorching hot, cold seeps leak dense hydrocarbon-rich fluids, primarily concentrated methane gas, slowly out of the seafloor at ambient, freezing deep-sea temperatures.
At the Freya site specifically, researchers documented massive, continuous methane flares rising over 3,300 meters straight up through the water column. These hydrocarbon seeps originate from ancient thermogenic gas and crude oil deposits trapped deep within Miocene-aged sediments.
And the craziest part of the Freya discovery to me is the biology. The chemosynthetic tube worms and the deep-sea amphipods they found living at these freezing cold seeps actually share deep, undeniable evolutionary genetic links with the organisms found at the high-temperature Arctic hydrothermal vents miles away.
It shows that there is this complex, totally unexpected ecological highway connecting these extreme disparate habitats across the vast dark abyss. The resilience and connectivity of deep-sea life constantly challenges our assumptions.
And the discoveries keep getting weirder. In 2025, an expedition operating offshore Papua New Guinea discovered an unprecedented hybrid geological environment. For the very first time ever documented, researchers found scorching, highly active hydrothermal vents and freezing, bubbling methane seeps coexisting side by side in the exact same field.
It is a geological pairing that scientists thought was practically impossible, and yet it is actively fueling a vibrant, highly diverse, overlapping chemosynthetic oasis of deep-sea mussels, giant tube worms, and entirely unknown species of sea cucumbers.
Okay, those discoveries are amazing, and they rewrite the textbooks. But we absolutely have to talk about the most explosive, highly debated, and controversial scientific paper published recently, the discovery of dark oxygen.
Ah, yes. You are referring to the highly polarizing paper published in the journal Nature Geoscience in July 2024.
I read the summaries of this, and it genuinely broke my brain. The study claimed to have documented the active production of oxygen gas at the very bottom of the Pacific Ocean, roughly 13,000 feet below the surface.
Yeah.
This is far, far beyond the reach of any solar radiation, meaning photosynthesis is completely physically impossible.
During a series of experiments using sealed benthic chamber incubations down in the polymetallic nodule fields of the CCZ, the sensors observed oxygen concentrations actually increasing over a 2-day period in total darkness.
The authors of the paper proposed an incredibly radical, paradigm-shifting hypothesis to explain the sensor data. They suggested that the polymetallic nodules themselves, those millions of years old metallic rocks targeted by the miners, act as natural geological geobatteries.
Their physical measurements of the rocks revealed unexpectedly high natural voltage potentials on the surfaces of these nodules, sometimes measuring up to 0.95 volts. They argued that this natural electrical charge, generated by the mixed metals in the rock interacting with the salt water, was just barely sufficient to spontaneously split the seawater molecules via natural electrolysis.
They were claiming the rocks were literally generating pure oxygen from the surrounding water in absolute darkness. Okay, I have to pause and push back incredibly hard here, and I know from reading the responses that I am not the only one. Splitting water molecules with rocks. That sounds exactly like a perpetual motion machine.
It does sound like magic.
How does a rock sitting in the mud constantly generate enough electricity to split water without draining itself? How does this hypothesis not violate the fundamental laws of thermodynamics? You cannot just conjure energy from absolutely nowhere.
You are loudly echoing the fierce, highly rigorous skepticism of the broader geochemical and biological scientific communities. The academic backlash to that paper was immediate and severe.
Subsequent papers and rebuttals argued exactly your point, that the proposed mechanism of continuous, naturally occurring electrolysis violently violates both the first and second laws of thermodynamics unless there is a massive, unknown regenerating energy source constantly recharging the nodules.
Many leading experts have pointed to potential experimental artifacts as the vastly more likely mundane culprit. They suggest that the dark oxygen readings were simply flaws in the calibration of the highly sensitive optode sensors, or oxygen that was accidentally trapped in the materials of the incubation chambers themselves, slowly outgassing into the water.
So, the scientific consensus is heavily leaning toward it being a busted sensor or a flawed experiment rather than magic electric rocks. But just for the sake of scientific wonder, let's play devil's advocate. What if the sensors were right? What if the phenomenon is actually true?
If the dark oxygen phenomenon is eventually validated by rigorous independent replication and proven to be a real thermodynamic reality, it is nothing short of earth-shattering.
It fundamentally suggests that aerobic life in the deep-sea animals that require oxygen to breathe may be partially or locally sustained by geological electricity rather than atmospheric mixing. It would prompt an immediate, radical rethinking of the geochemical conditions necessary for the original emergence of life on early Earth.
Furthermore, it dramatically increases the statistical odds of finding complex aerobic alien life on completely ice-covered ocean worlds in our solar system, like Jupiter's moon Europa or Saturn's moon Enceladus, where solar energy is non-existent but rocky, metallic cores interact with saltwater.
It is the ultimate high-stakes scientific mystery. It could just be a broken thermometer, or it could rewrite the origins of life in the universe, which perfectly sets up the final, overarching question of our discussion today: the roadmap.
How do we actually answer these seemingly impossible questions?
Exactly. How do we definitively prove dark oxygen, or map the dark taxa, or find the colossal squid before the deep-sea mining conglomerates destroy the evidence? We clearly need tools that go far beyond a ship blindly dragging a net, and far beyond dropping a tethered, noisy camera that terrifies the wildlife.
We do, because as miraculous and revolutionary as environmental DNA is, we must acknowledge its operational limits. A genetic sequence pulled from a water filter beautifully answers what kind of animal is in the area, but it severely struggles to answer how many of them there are or in what physiological condition they exist.
Determining absolute population abundance, or actual physical biomass strictly from genetic traces, remains mathematically elusive because, as we discussed, the shedding rates and the taphonomic degradation rates are so incredibly variable depending on the species and the current.
Right. A microscopic strand of DNA floating in a tube cannot tell a marine biologist anything about the organism's actual behavior, its specific life stage, or its ecological health.
It can flag the presence of a dark taxa sequence, but it cannot tell us how a newly discovered blind crustacean navigates the toxic chemical gradients of a boiling hydrothermal vent, or how a colossal squid coordinates its massive tentacles to ambush a toothfish in the dark. You need to actually observe the ecosystem functioning.
To move beyond isolated static genetic sampling and capture the dynamic reality of the deep ocean, the scientific roadmap for the next decade, which aligns closely with the United Nations Decade of Ocean Science and the ambitious Seabed 2030 mapping project, requires a massive shift toward multimodal, highly autonomous technologies.
We are actively moving away from human-occupied submersibles and ship-tethered ROVs and transitioning heavily toward long-range autonomous underwater vehicles, or LREUVs.
These are basically the advanced artificial intelligence drones of the deep sea. They aren't tethered to a ship. They are pre-programmed to operate independently for weeks or months at a time.
They are equipped with advanced 3D acoustic multi-beam sonars and sophisticated, highly sensitive chemical sensors, capable of mapping the complex topography of the seabed and dynamically tracking the chemical plumes of undiscovered hydrothermal vents entirely without direct human intervention.
But from everything I read about the future of marine tech, the absolute holy grail of this entire autonomous roadmap is a device called the ESP, the Environmental Sample Processor.
The ESP represents a monumental, paradigm-shifting leap in biological ocean monitoring. It takes the laboratory out of the university and puts it directly into the abyss.
The way I visualize the ESP is like having an advanced NASA Mars rover. But instead of slowly driving across a dry red desert to zap rocks with a laser, The ESP is a fully automated, miniaturized robotic laboratory swimming silently through the freezing bathypelagic zone.
It physically filters the water, chemically extracts the eDNA, uses internal microfluidics to sequence the genome in situ, right there in the freezing dark, thousands of meters deep, and then it surfaces to beam high-resolution, definitive taxonomic data via satellite back to scientists on shore in absolute real time.
That is exactly the capability it provides. It dramatically exponentially increases both the spatial and temporal resolution of deep ocean monitoring. Through the deliberate convergence of real-time environmental DNA sequencing, advanced acoustic biomass modeling, and complex AI algorithms integrated directly into these quiet, autonomous platforms, humanity is finally developing the technological capacity to gently turn on the lights in the abyss.
But the tragedy of our timeline is that those lights are finally turning on just as the industrial bulldozers are warming up their engines. That is the core, unavoidable tension of everything we've explored today. The brilliant, desperate race to decode the taxonomic dark matter of the sea is running in a dead, parallel heat against a highly capitalized industrial race to exploit it for profit.
The ultimate scientific and moral question for the coming decade isn't just a matter of what alien wonders are hiding down there in the dark. It's a question of whether the global scientific community can mobilize this incredible technological roadmap fast enough to comprehensively map, understand, and protect these fragile ecosystems before they are irrevocably altered or entirely erased by deep sea mining.
It is a profound civilizational responsibility. Whether we recognize it or not, we are the custodians of a massive, vital planetary system that we barely comprehend. The deep ocean is not a barren void. It is a vital sanctuary of ancient evolutionary lineages, bizarre and instructive physiological adaptations, and incredibly critical climate-regulating machinery that our own atmospheric survival depends upon.
And as we wrap up our exploration today, I want to leave you, the listener, with a final provocative thought to ponder. We talked extensively about the extremes of survival gigantism, glowing twilight fish, and thriving in toxic, freezing darkness.
What if the definitive key to humanity surviving the next chaotic century of terrestrial climate change? Perhaps a highly resilient novel gene for drought-resistant crops, a revolutionary new pharmaceutical compound to fight antibiotic resistance, or a radical, hyper-efficient carbon-sequestering enzyme is currently locked quietly inside the DNA of a bizarre, gelatinous creature floating in the Twilight Zone.
A creature that we, in our rush to exploit the Earth, haven't even bothered to name yet.
Stay infinitely curious about the massive, invisible world beneath the waves. Thank you for exploring the depths of knowledge with us on Audio Notebook. Until next time, keep looking deeper.